Hello, everyone. Good morning, good afternoon. We are a minute past the hour, so we're going to get started with the webinar today. So my name is Amitava Mitra. I'm from Sandoz, Novartis, and I will be the moderator for the webinar today. So welcome again. As you can see on the slide, I hope everybody can hear me okay and see the slides that's being projected. If not, please speak up or post a comment on the discussion board that you must have on your viewing screen. So as you can see, the title for today's webinar is Grill the GastroPlus Gurus. Arlene, if you could go to the next slide, please. Thank you. So we have four stellar speakers today: Michaela Reddy, Phil Bransford, Wei Zhu, and Mike Bolger. And I'll introduce all of them as we go about the webinar. But as you can see from the years of experience, I did the quick math, and looks like we have 50 plus years of combined experience. So I think this is a great opportunity to learn from these folks. And I suggest that you grill them well during the Q&A session. So before we get started, some housekeeping. So we have a slightly different format for the webinar today, and it's the first time we are exploring this. So if something doesn't go well, so apologies in advance, and we would love to hear your feedback on this format. So as you can see, we have four speakers. So we'll have, each of them will have roughly 10 minutes to talk on their topics, which are listed on the table. And we will allow 2 brief questions after every talk. And I suggest that you keep the questions to clarifying questions only. And if we stick to the timing, we should have roughly 30 minutes for a detailed discussion at the end of all 4 talks. And that's when we can have a robust discussion or detailed Q&A on your queries. The webinar is being recorded, and it will be available to all user group members in future. And regarding asking questions, we already have a few questions that were posted ahead of the webinar. So thanks to folks who did that. They're pretty fantastic questions, and I'm sure we'll have some good discussions on those questions. And if you want to ask questions live during the webinar, you can do it using the questions panel on the control panel that you have. Or you can just raise a hand by pressing the hand raising icon and ask the question live through your phone. So with that, we'll get started with the webinar. But before we do, we'll have a short poll to begin with. So Arlene, if you can bring the poll up, please. And you'll have 30 seconds to respond to this starting now. Thank you, Arlene. So we'll now start with the webinars. Our first speaker is Michaela Reddy. Michaela is currently a clinical pharmacologist at Autoit Biopharma, which is now a Pfizer company. She has been there for 6 years, supporting discovery through first-in-human dose predictions and also late-stage projects. Prior to this, Michaela was a modeling and simulation scientist at Roche. So with that, Michaela, the floor is yours.
Okay, thank you, Amitava. Can you hear me?
Yes, I can, loud and clear.
Okay, great, thank you. So I wanted to thank all the attendees and just emphasize that all the panelists today are members of the GastroPlus user group steering committee, and we have an active group on LinkedIn. And we are always looking for new members. I'm the chair of the steering committee, and if you just contact us through the LinkedIn group and you're interested, we'd love to have you. So after that plug, next slide, Arlene. Okay, so I just wanted to emphasize that parameter sensitivity analysis is an important thing that we all need to do. And with modeling, starting in discovery projects, and then ideally we do modeling all the way through first in human, and then refine the model once we have the human data. I think sometimes people talk about parameter sensitivity analysis as if it's I played around with the model parameters, and I think that's really the wrong way to look at it. And I think that the recent PBPK guidance documents from the FDA and EMA kind of support that, and they emphasize that it's important. And just for information, I kind of go back and forth here. Parameter sensitivity analysis is looking at how important a specific parameter is for simulation results. And then also I touch on sensitivity analysis, which is, in my view, more if there's a mechanism that would be incorporated or not incorporated, that to me is a sensitivity analysis, and they're related and they're both important. So I kind of touch on both today. But anyway, the FDA and the EMA have both said that they think it's important, and they want to see that as part of submissions when you have regulatory submissions with PBPK modeling. And in fact, just based on my experience, I've actually had requests for parameter sensitivity analyses and sensitivity analyses from both the FDA and the EMA with regulatory submissions. So, the guidances are consistent with the feedback we've gotten from them. Next slide, please. So I guess as a beginner, maybe the first question you might think of is well, what should I be looking at even? And that's kind of beyond the scope of what I'm trying to present here today, but I thought I could point out this paper that might be a useful reference. This is actually another GastroPlus user group product. A bunch of us from the user group got together and put this paper together with practical examples. I think for uncertainty analysis, when you have limited confidence in some input data, that might be something you'd want to examine. BCS class should play a role in your determination of what you need to look at. It could be for the absorption model. It could be for the the PK piece of the model. Anyway, this paper will be a useful resource for you in determining what to look at. Next slide, please. Okay, so I'm focusing mainly on oral absorption of a BCS class 2 molecules today. I'm presenting discovery examples, but I'm also going to speak to a clinical example at the very end. So this is a BCS class 2 molecule, and GastroPlus has this useful capability of doing these parameter sensitivity analyses. It's not the only way to do it. I've definitely presented results in tables. I've presented CP time plots with a range of parameter values, but this is a useful plot to generate, and so I'm going to focus on this. And you can see these spider plots. I think a lot of us GastroPlus users, we know what these are, and it's clear to us. But if you look at the next slide. So what I have actually found is that it might be useful to us, but to other people it can be kind of confusing, because when you show all the curves on the spider plot, it just shows the first clicked box on the x-axis. So one of the things that I like to do, and this is a tip for beginners, I guess, is unclick them all except for one at a time, and then copy multiple versions of the plot into the presentation. So the top left is the spider plot, and what I do is, in the top right, that's just the particle radius clicked. Bottom left, that's just the reference solubility clicked. Bottom right is just the log D clicked. And then once you do that, you can kind of put red curves pointing out the region where the model is sensitive, and so it kind of increases the utility for communicating with teams on key issues. And the other thing I wanted to point out is normally when I present human PK projections, I do it to a room full of people, and sometimes people don't feel comfortable asking questions in that environment. Recently, I met with two scientists, and one of them was a chemist, and he said to me, I've seen a lot of these plots, and I never really had anyone explain to me what it meant. And he said, can you walk me through it? And I'd been making these presentations for 5 years, and I guess I just got used to kind of throwing the plot up there. So it's just a reminder that, that old adage that you should walk people through complicated plots every time is true, and it's just something to keep in mind. Next slide, please. So another tip for beginners, I think most people will show a single parameter sensitivity analysis, but oftentimes the results of the parameter sensitivity analysis might depend on another parameter. So you can do a three-dimensional parameter sensitivity analysis. That's a capability of GastroPlus, and sometimes it's useful. What I like to do is look at two situations and that I think might be interesting, and I'll put up sensitivity analyses for each. So you could do it for a human and a rat, or a fed and fasted state, or a typical subject and a poor metabolizer. The example that I've shown here is kind of a classic example, is for a BCS class 2 molecule, and I think especially in that case, or a BCS class 4, it would be really important to look at a low and a high dose to really understand which model inputs might be important. Okay, next slide, please. So in a tip for more experienced users, I think it's really important to think carefully about your parameter sensitivity analyses results. And sometimes we look at parameters, and it's legitimate to do so, but you just have to remember that sometimes, like, for example, the C log P affects multiple aspects of the model, right? So this is just one example, but I'm sure there's others. So the C log P affects the absorption prediction. It affects the clearance prediction if you have done the protein binding value adjusted for the binding to plasma lipids. And it also adjusts the tissue to plasma partition coefficients. So it can affect multiple aspects of the model. So it's not that you shouldn't look at it, but you just need to think carefully how to do it and think it through. So if you were going to look at C log P as one important model parameter, then you might want to do it, just look at your absorption prediction instead of look at your AUC or your Cmax, because those would show the multiple aspects kind of confounded. Next slide, please. So as another tip for more experienced users, I like to look at things from a PK/PD perspective. Just looking at the extent of absorption, I think that's important. You want to have a molecule that's well absorbed. But if you're trying to develop a molecule for kind of a high bar with your target, you're really being aggressive with how much coverage of the target you're trying to get, like for a Cmin, then I think you need to kind of go beyond the typical, will absorption be sufficient, and think in terms of the absorption window as an example. So in this case, this is a molecule where the measured log D was 3.1 and the calculated was 3.1. I mean, you can have problems with in vitro data. Maybe the in silico value is more representative of the compound properties in this case, but I think that range of 2.1 to 3.1 is probably realistic for the molecule. And for 2.1, the plasma concentration at 24 hours wasn't high enough, and at 3.1, it was high enough. The parameter sensitivity plot suggests that this is not a really important parameter, but it's affecting multiple things, and it's also affecting that absorption window. So it turns out for a Cmin, it's actually critical. And so the prediction of colonic absorption in silico, to me, is one of the things that I have less confidence in, and so I want to do more checking against preclinical species and think more carefully about that. So in this particular example, it's just important to think about it from a PK/PD perspective and really look at the preclinical data and verify the model in terms of that regard. And if you just looked at the parameter sensitivity analysis plot, for the measured log D, I think that aspect would have gotten lost. Next slide, please. Okay, so my final tip is about considering including data with your sensitivity analysis when possible. So this is just an example, another BCS class 2 molecule, and the in silico pKa was 3.7. I had some uncertainty around that, and the other thing is that the mouse model was non-fasted, so you could have some animals that are fed and some that are more similar to a fasted state. So this is just one example where I show the data like against simulations for both the fed and fasted state. And it seems to provide some insight. So whether that means, you want to measure the pKa or, it just gives you enhanced understanding that you're going to have some variability in the Cmax. I think this was useful. Another example is I had submitted some PBPK modeling to the EMA for some regulatory, applications. And they had come back to me and they said, we actually don't like the value you chose for permeability. We want you to do some sensitivity analysis and try to convince us that it's correct. And I'm paraphrasing here. And so what I actually wound up doing is I did some parameter sensitivity analysis, and I plotted simulated versus observed Cmax across doses for a range of permeabilities and for a range of fed and fasted states, because the patients were instructed that they could eat or not eat, and basically they shouldn't have to worry about food. And I was able to show them that the model parameter that I selected was reasonable and the most consistent with all the data. And so I had to do those simulations in GastroPlus, and then what I did was I took the simulations and the data in Phoenix and constructed those plots. Okay, well, that's my last slide. So I think I have a summary on the next slide. So parameter sensitivity analysis done well is really a critical part of modeling and simulation results, and it's important to think about it and do it carefully. And I suggest considering whether multiple cases might be useful, whether a parameter might impact multiple aspects of simulation results, and whether data could be compared to the results for additional insight. So thank you.
Thank you, Michaela, and right on time too. So we do have time for two quick questions. I see there are two folks who have raised their hands. Franco, do you want to go first? Arlene, do you have to unmute him? Okay.
Okay, can you hear me now?
Yes.
Okay, a quick question for Michaela. You were presumably doing the last example in mice for a compound that was advanced toward the candidate level. That's a general question. Why do we keep using in silico? You should have had the pKa measured.
It was actually an early stage molecule.
Oh, it was, okay. So you were doing a prediction. Okay, then my question is answered. Thank you.
Okay, sure.
And we have Raj Kumar. I think you are unmuted. Well, I guess not. Are there any other questions? Okay, if not, then we'll move on. Thank you, Michaela.
Thank you.
So our next speaker is Phil Bransford from Vertex. So Phil supports API form and formulation development at Vertex, and he has a PhD in biological engineering, and he's going to talk about solubility model calibrations. So Phil, the floor is yours.
Thank you, Amitabha. So today I'll be talking about a pretty standard procedure when building a GastroPlus model for a small molecule that's delivered orally, and that's calibrating the model with biorelevant fluids, simulated intestinal fluids. And what I found in my experience is there's a potential pitfall in doing this that I'd like to make everyone aware of, and also at least one proposed solution to it. So that obligatory motivation slide. So why do we use biorelevant media in building oral absorption models? And it really comes down to the property space that a lot of the new drugs that come out these days live in. So over time, new chemical entities have sort of migrated in this chemical space to a high log P or lipophilic space, and also high molecular weight. So what comes with that is compounds that are developed these days, they tend to be poorly soluble, and they also tend to interact with biomicelles, which are the endogenous surfactants in the GI fluid. So on the right is just a plot showing solubility data for 4 different compounds as a function of the bile acid concentration, and this is all at a constant pH. So what you can see is the solubility increases with the bile acid content, and it's because these compounds are somewhat lipophilic. And for each compound, the slope's a little bit different, but they're all positive. So there's this positive interaction between the API and the biomicelle. So GastroPlus, it has a tool for modeling this effect. You've probably seen it before if you use biorelevant solubility in the model. And the way it's used is you enter fasted state, simulated intestinal fluid, fed state, simulated intestinal fluid, and you click on the little fit button, and it fits the model. And while I'm talking about this, I should mention just one point that's important to keep in mind. The values for the pH and the bile acid content, those should be specified based on your experimental conditions, and I wouldn't use just the default values there. So sometimes the pH drifts in the experiment, so I always use the final pH of the experiment. And the other issue is depending on the FedSIF recipe, whether you're using version 1 or version 2, the taurocholate concentration may be different. So it's important to use the correct numbers when doing this calibration. And so what's going on under the hood when we do this fit? It's fitting a model, of course. So the solubility is considered a function of two variables, the pH and the concentration of bile. And it can be written in this separable form where the first term is just the pH solubility curve, absent any surfactant. And the second term shows this linear dependence of solubility on bile acid concentration. So one way to look at this is, here's an example compound. This is from the demo database with carbamazepine. So the aqueous solubility, at pH 6.8, it's fairly low. That's shown on the left. You can see the linear increase in solubility with the bile acid concentration. And this model works quite well for this compound. So it's always a good idea to make sure that when you do this calibration, that the actual predictions match what's experimentally measured. And the approach I use is to go and toggle over to the physiology tab and temporarily change the pH of the duodenum and change the bile concentration as well to match what was used in the experiment. And then just verify that this predicted value for the solubility in the duodenum matches what was measured. And same thing for the fed state. So in this case, the numbers are spot on, the model and the prediction. So I'd feel confident in going and predicting food effect or doing all sorts of other types of modeling. But sometimes the model can fail. So here's an example where similar compound, but as you can see, the fasted state data is about 22 microgram per mil, whereas the prediction is 80. So it's off by almost a factor of 4. And you can compare that with the fed state, which is about, it's pretty close. It's the first sig fig is right. So in this case, the model is not doing so well. And I've encountered this quite a bit recently. I think more often because we have switched our media recipe from a version 1 recipe to a version 2 recipe. So it's a good idea to check for this issue every time you build a model. And I'll show you one way to try to deal with this type of problem. So the model isn't always, a linear model isn't always appropriate for your data. Sometimes we can get what I call a nonlinear behavior. So on the right is showing the concentration, the solubility as a function of bile acid concentration when it follows a nonlinear model. And in this case, it actually increases as the bile concentration squared. So if you fit a linear model to this plot on the right, you'll dramatically overestimate the fasted state solubility. You'll be okay with the fed state, but this can have, as I'll show later, a pretty big impact on the model predictions. So what I would do if I plot these data and I see this behavior, is I would go and try to figure out what is that correct scaling factor. Should that n be 1 or 1.5 or some other number that gives me a good fit? And then when you go back and try to calibrate the model, instead of putting the bile concentration, you'd put in the bile concentration raised to that power. So in this case, the power was 2. So instead of 3 millimolar for fasted state, it's 9. Instead of 15 millimolar for fed state, it's 225. And in this case, when you check the model to see if it's fitting it, it's spot on. So we've got a nonlinear model that can predict the solubility quite well. But of course, GastroPlus has a linear model. You can't get the source code and recompile it, so you have to basically trick the software into implementing a nonlinear model. And the way you can do that is actually pretty straightforward. Is instead of using the standard bile salt concentrations in the physiology tab, is you just replace them with numbers that have been raised to the correct power. So in this case, the correct power is 2. So what GastroPlus will do, it'll grab the number from this column in the table and it'll stick it into the equation, and it'll be implementing a nonlinear model using a linear equation. And you could do this with other types of functions too. It could be a step function or piecewise linear. Really, it just depends on what you enter into this column. And as far as I know, the bile salt concentration is not used anywhere else in the model, so doing this kind of change of variables shouldn't affect other parts of the model. And so what's the impact? So if I use those data and fit a linear model and I have a bad fit, it might come up with this prediction, which is shown on the right, which is that there's no food effect for this compound. The fasted and fed PK are nearly identical. Whereas if I use the nonlinear model, which fits the data quite well, it shows there's going to be this profound food effect. And I've encountered this in my own experience, where in one case we predict no food effect, and in another case we predict a positive food effect. And this can have pretty significant implications on your clinical design if you're going into phase 1. You may want to go into phase 1 with an enabling formulation in your back pocket versus proceeding with a conventional formulation. And there are other approaches to calibrating the model as well when the standard linear model doesn't fit. So one proposal was to fit separate solubilization ratios for fasted data and fed data. This can work just as well as using the nonlinear model. And the other approach, you just turn off the bile acid correction entirely, and you input the fasted state data in the reference solubility entry, and you put the fed solubility in the reference solubility data, and just have two different models. With this case, you lose the bile salt gradient in the GI tract, so you might overestimate the amount of absorption, especially in the distal part of the small intestine and the colon. So the takeaways are, it's always a good idea to check to see that the model is actually predicting what was measured experimentally. Be mindful that sometimes you'll see nonlinear behavior. It could be because the media recipe has a critical micelle concentration and there's no interaction between the API and bile at low concentrations, but a significant one at high concentrations. And then the last is you can trick GastroPlus into modeling this nonlinear behavior by modifying the physiology inputs. So with that, I'm happy to take a couple questions.
Thank you, Phil. I don't see any hands raised, but we do have time for 2 quick questions. Okay, so I guess in the interest of time, we will move on. I do see one hand raised, Saurav.
Hello, sir. Good evening.
Hello.
Yeah, go ahead, please.
Sir, what will be the effect of other GI tract contents?
Yeah, so the question about what's the effect of other GI tract contents. So in the GastroPlus model, which we're kind of limiting our discussion to, we only have the effect of bile acids and pH.
Yes.
There's obviously other goop in the small intestine, and there's also different components in the simulated fluid. But at the moment, the assumption is the dominant effects are pH and bile acids. So I think it's still okay to use a model like this and just realize that you're not including everything that's actually there when you build a model, and it could be the reason why a model doesn't predict well in some instances.
Okay, sir. Okay, sir. Thank you.
All right, thank you, Phil. So with that, we'll move on to the next topic, and the speaker for this particular part of the webinar is Wei Zhu. So Wei is currently working in the Biopharmaceutics and Specialty Dosage Form Group at Merck, and he has been with Merck since 2008. He has a PhD in pharmaceutics from University of Houston, and his main research interests are in vitro, in vivo, and in silico Biopharmaceutics evaluation of oral and alternate route dosage forms. And Wei is going to talk about lower gut absorption. So Wei, the floor is yours.
Thanks, Amitabh. Can you hear me?
Yep, loud and clear.
Okay, thank you. So my name is Wei from Biopharmaceutics and Specialty Dosage Form Group in Merck. And today I will share some of our work experiences on lower gut absorption model as part of the user group webinar today. So I will follow these outlines. I'll start with general considerations, assessment of lower gut absorption and MRCR feasibilities from biopharm perspective, such as BCS, in vitro ex vivo, in vivo models, and briefly describe our general experiences on GastroPlus lower gut model in Merck, followed by the development of a customized lower gut absorption model with dog-to-human translations and a case study. Next slide, please. Right, so there are multiple factors affecting the bioperformance of our CR/MR formulations, such as physical chemical factors, formulation factors, and physiological factors. For example, the solubility capability of the drug in lower gut may be impacted on lower luminal volumes and the lower bile salt concentrations compared to the upper GI. From biopharm perspective, BCS is probably the first line of the differentiator to assess the feasibility of MRCR formulation, such as for compound like BCS1, 2 drugs are usually better candidates due to potentially high permeability in lower gut. And there are various in vitro cell-based and membrane-based models to assess the absorption of the drug in lower gut, as listed here. However, the physiological differences need to be considered, such as enzyme and transporter expression levels, difference on tight junctions in the lower gut versus in vitro models, etc. So the dog models, as we are going to discuss today, in conjunction with in silico approach, is actually the dog model with retrograde catheter, which directly introduce the drug to the intestinal region. As you can see, the diagram actually on the bottom right side of the slide, with the advantage of taking into account of both permeation and the solubilization of the drug. Another benefit of this model is that it allows a direct comparison to oral administration of the drug in the same dogs with possibly a crossover PK evaluation. Next slide, please. So from GastroPlus side, there are quite a few publications on discussing the modification of the colonic model, such as adjusting the volume, etc. According to our modeling experiences in Merck, the default settings may oftentimes overestimate or underestimate the absorption of a certain drug in lower gut. So the idea of this work presented today is based on the fact that the default ASF values in GastroPlus were derived by finding the best compromised values across a large number of drugs. So they should not be expected to apply to all of the compounds. The absorption scaling factor, which is also called ASF in GastroPlus, can be significantly different if the permeability is significantly different in the different regions of the GI tract due to, other factors such as ionizations, of the or the presence of the transporters, things like that. So, so here in this presentation, so I will describe briefly how we use the customized ACAT model to estimate the lower gut absorption and further translate the absorption model from dogs to humans. So the development of this lower gut absorption model is a stepwise process, starting from building oral absorption models based on the dog IV PO PK data, and calibrate the lower gut absorption settings in dog model against the clonic dose dog PK data with the dog model of the retrograde catheter method that described in last slide, and also validating the absorption settings in the dog models against the CR formulation oral PK data. And then we extrapolate the lower gut absorption settings in GastroPlus from dog model to human model. So once we have these, we have the model ready. We are ready to predict the PK profile of a certain CR prototype formulation in clinic with calibrated human model. Next slide, please. So this is a relatively busy slide, so I'll walk you through this. So this is kind of like a case study for compound A, which is BCS2, borderline BCS1 compound with decent intestinal solubilities. So due to narrow therapeutic window, there is a desire of developing a CR formulation. So the good news is that the regional bioavailability study in dog model showed actually a good colonic absorption of this drug. So after we have the oral dog PK model ready, as you see in this step 1 here in the slide, here we modify the default ASF model to reflect the condition of the colonic dosing of a solution formulation, either as a 1-compartment or 2-compartment lower gut model. So in order to make it happen, the transit time and the ASFs were set to 0 for stomach and all upper GI compartments. The basis of the 2-compartment model is actually a theoretical SAV models in dogs, while in 1-compartment model, the transit time were added together by the default transit time in cecum and colon in 2-compartment model default settings. The default ASF settings were actually also adjusted accordingly based on the definition of the ASF in theoretical SAV model, which takes into account of the surface area and enhancement factors and the radius of the lower gut. So once we have this baseline model set up, so we optimize both model 1 and model 2 against the dog PK data with clonically dosed formulation as shown here in step 2 by adjusting the lower gut ASF values in each model. We obtain the correction factor for human model in step 3 in both 1-compartment and 2-compartment models by calculating the ratios between the optimized dog ASF against the default dog ASF. Next slide, please. So once we validate the absorption settings in the dog model against the PK data from the chronically dosed solution formulation and also the oral PK data with CR formulation in dogs, we move on to set up the human absorption models in GastroPlus. So we also modify the default model in GastroPlus by using the previously obtained correction factors in last slide in the dog model and extrapolate to the lower gut ASF settings in humans. So similar to the dog model, the basis of the 2-compartment model is a theoretical SAV model in humans, while in the 1-compartment model, the transit time and the default ASF settings, the ASF settings were adjusted accordingly based on the definitions in this model, which takes into account of the SEF of the ascending colon, SEF of the jejunum, and the radius of the lower gut. Next slide, please. So once we made this customization of the model, we made PK predictions of 3 CR tablets of compound A with different release rate. On left side is the in vitro release rate of the formulations, and on the right side is the simulation results of the PK profiles in either 1-compartment or 2-compartment models at 2 milligram dose. So the prediction results showed the exposure is actually in general in line with the observed data in both models, although you can see there is a slight difference on the simulation outcome in both models, which is expected. Next slide, please. So I think this pretty much is just a quick summary and takeaways of my presentation of this, my part of the presentation. So calibration of the absorption model on the lower gut absorption settings with preclinical data showed promising outcome to support CR feasibility assessment in humans. And however, I think more importantly, as a modeler, on the call, I would say the physiologically based PK and biopharm modeling is a data-driven process. So it is critical to check and refine the model appropriately at different stages of drug development using available data with properly designed experiment for parameterization of the key aspect of the model. All right, so next slide. I think that's pretty much concludes my part of the webinar presentation. Thanks for your attention, and please let me know if you have any questions or comments. Back to you, Amitabha.
Thank you, Wei. Great talk. So I do see a few hands raised. So Saurav, do you want to go first? You have to unmute yourself, I guess. Are you still there? Okay, there you are.
Sir, what about other models, other animal models? Can we go with other animal models to mimic the condition of human model?
Well, I think there's certainly other possibilities. For example, rat intestinal perfusion models and others, even like the ex vivo models. But I would say that the work presented here is pretty much one of the approaches that we are using to optimize the lower gut absorption settings. And certainly that there are other models that you can have a look. I think it is a possibility, but you will have to keep in mind the species difference in the local transporter expressions, absorption capacities, tight junctions, the differences over there. And also, like if you look at the in vivo versus ex vivo conditions, there's some other things, additional considerations to be think about.
Okay, sir. Can we say the dog model is best fitted to the human model among other models?
Well, I wouldn't really make this claim for all of the compounds and all of the CR/MR formulations. We know that there is a difference in the prediction performance on the matrix-based versus the osmotic-based model formulation for the translation between dogs and dogs to humans. But I wouldn't really make a claim to say that dog model is applicable is the best model for all of the projects.
Okay, sir. Thank you.
Okay, maybe one more. Natalie, do you want to go?
Hi, thank you for a very interesting presentation. My question is extremely practical. When you estimated or optimized your ASF for your dog model, you set up your transit time to 0, which I understand, but you also set up your other ACF to 0. Is it really necessary, or is it just an habit?
Well, I think it's probably technically it is not that necessary because you have already have the transit time to 0. But just to be just to make sure that we are really exclude all the absorption potentials in the stomach and the upper GI. So I pretty much set everything to 0. Actually, practically when you set up these ASF values, customize these ASF values, you cannot set to a 0 value. It has to be a very low values that if you want to go there. But it is something that I usually set as 0.0001, things like that. But I think the idea is just to make sure that there is no absorption from the upper GI level.
Yeah, I guess it makes sense because also when you set up the transit time to 0, you can't set it up really to 0. You've got to use a very, very small number as well. So potentially you could still have some absorption. Thank you very much.
You're welcome.
All right. Thank you, Wei. Franco, I see you have a hand raised too. If you don't mind, if you can hold on to your questions till the Q&A, we're just going to move on in interest of time. So our next speaker is Mike Bolger. A lot of you probably have already interacted with him as you work with different tools from Simulations Plus. So Mike is the chief scientist at Simulations Plus, and he has been there for 20 years. In fact, he programmed the first version of GastroPlus in 1997. And prior to Simulations Plus, Mike was a professor of pharmaceutical sciences at University of Southern California School of Pharmacy. So with that, Mike, I'll give you the floor.
All right. Thank you for the introduction, and thanks to Michaela and the user group for inviting me to give this talk. This is something that we actually built into the very first version of GastroPlus, but doesn't show up too much in our workshops or other things. And so 23 years later, it's kind of nice to be able to discuss this in case you find useful aspects of it. The idea here is that we have a lot of ways to automate the procedures that you would do in GastroPlus when you have to do a repetitive task. You may want to optimize the intrinsic clearance for 23 different records. And if you want to just sit there and use your mouse to, press the, modules, optimization, set parameters, pick the parameter, and then go back to the start screen and press optimize, and then wait for it to optimize, and then do the next record and go on to the next thing. You're sort of limiting the amount of time you can use your brain to think about the actual problem. And so the whole idea of the keyboard shortcuts and potential automation is that you are able to set that up and go to lunch and come back and everything's done. In GastroPlus, we have keyboard shortcuts that are visible. And these are visible through pressing the Alt key and then the underlined letter that is involved. We have some invisible Alt keys that, I'll explain why they're invisible. We have some invisible control keys. In the earliest versions of Windows, these were all visible, but as Windows evolves, they have sort of moved away from making Alt keys and keyboard shortcuts accessible. We're going to talk a little bit about tab stop order, so that if you press the tab key frequently, you'll go from one field to the next, and a shift tab will just do the reverse. Then we have several things in GastroPlus that have sort of eluded our programmers in coming up with consistency. Most of the time, an Alt-O will be used for OK, but you'll find that under cancel, it's Alt-N or Alt-C, and in some cases, Alt-C for close. One of the things that is useful when you're sitting in GastroPlus is to be able to sit on one of the tabs, like the pharmacokinetics tab, and then move between records so that you can see visually if there are any fields that are changing in terms of volume of distribution or clearance. Viewing one tab, activating a button on a different tab, and then finally I'll demonstrate a program called Autoit. It's a free download. You can just Google Autoit, and this is a very useful tool to set up a script that would automate all of these Alt keys. So visible Alt keys. First of all, we're going to observe one letter of each tab that is underlined. So if I move over to Gastro, in the normal situation, GastroPlus would not be showing any underline under the file key. And once you press the Alt key, then the underline appears. So there's the underline, and we can see that Alt F is the key for the file drop-down menu, E is for the edit, D is for the data, etc. Once you continually press the Alt key, it cycles back and forth. Say we want to initiate a structure import using the Alt keys. What we would do is press Alt F, and now you can see there are other menu items that are underlined. So the letter I for import would work, and then the letter S for structure. Now, to me, this is a lot easier than wasting your wrist mobility on trying to move through these menus. I've seen many people, fumble with that. So Alt F-I-S, and then you can pick up the information that's necessary, like an SDF file. And all of these steps can be automated because we have the Alt OK, Alt C, and we have tab stops. I'm not going to run through this particular one. Let's say that you wanted to see the OPD file, Alt F, L for load, and number 6 would be for the OPD, number 5 would be for the intravenous. So I'll just press the letters number 6, and up pops this form, and then Alt O to OK or Alt C to cancel. There are visible Alt keys on the Graph tab. So let's say I do a, start, and now I'm going to go to the Alt G right over here to get to the Graph tab. The latest version of GastroPlus, this is the first time that I think we've had Hai Ying in particular was doing the programming. These are all Alt numbers. So Alt 1 is a new plot, Alt 2 would be for absorption dissolution. So I'm just clicking the mouse right now. Alt 3 would be for CP time, Alt 4 would be for the regional distribution. If I move my mouse off there and hit Alt 1, then we get the new plot. If I hit Alt 2, we have to, of course, be on the form. Alt 2 would be for the dissolution, Alt 3 would be for the CP time, and Alt 4 would be for the regional distribution. So the same thing goes for 5 for save all data and save selected data. These are useful shortcuts that keep your hand off the mouse. You avoid carpal tunnel syndrome, and you can really move forward with your work. Invisible Alt keys. Now, these are ones that, for whatever reason, Windows has decided to eliminate. So even if we program in the underline K on the pKa table, the latest versions, well, not the latest, the way that Windows handles this is to never show that. Even if I press the Alt key, we don't see the K being underlined. So we call these invisible Alt keys. But if I press Alt K, then the pKa table comes up. And then I can press Alt N to cancel. If I press Alt Z, then the enzyme table comes up, and N to cancel. So you can see there's a lot of flexibility with regard to using these types of Alt keys, even if they're not visible. Let's suppose on the simulation tab, we have the Alt T to start and Alt P to stop, but we're not actually looking at the simulation tab. Maybe we're looking at the Graph tab, and I press Alt T to start. Well, now the simulation is running, and we don't have we got another meeting, but forget about it. And we could then, cycle between the various plots. Invisible control keys. Now, these are invisible because you usually don't see them when you're just looking at the screen, but we do have menu items that tell you what those control keys are. So for example, Control N would be for the next record, Control P would be for the previous record, Control F first, Control L for last, and Control G to jump to a particular drug topic. So if I'm in gastro and I'm on the compound tab and I press Control N, then I move to the next record and the next record and the next record, Control P, Control L for last, and Control F for first. Or if I press Control G, if I press Control G and type in any part of the drug name, then we're going to jump to that particular record. So here we just jump to ranitidine. Tab stop order is also useful. So if I were to go to the biorelevant solubility, I think, let's see, and start to press the tab key, tab moves now to the temperature, to the melting point, to the adjustment, and I can turn these things on and off with the space bar. Tab, 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 off, on, just the space bar will do that. And you don't need necessarily the mouse. On, off. And this just reminds you about the space bar, and we'll make, of course, these slides available. Now, this is the team that has been building all of the GastroPlus Visual Basic information. We will have another meeting next Thursday. It's just a bit of an advertisement for the GastroPlus users group, and we'll be discussing some of the latest progress on GastroPlus 10 or GPX. Okay, the last topic is to show you something about Autoit. So Autoit is a program you can download. It's free, and you can set up various scripts. So this is a very simple script. It just says, show me a message box, and have it say, this is my first script, and then use the text "Hello World" in the output. And so if you right click on an AU3 file and say run the script, then it pops up a message box and says "Hello World." Now, the next one is going to be a edit script, and we're going to basically open up, we're going to wait until we click on GastroPlus, and then we're going to activate the script. And the script will first open a message box and say that it's going to send an Alt-Y key. The next thing would be to send the Alt-Y key. So the exclamation point is the character Alt, and Y would be the character that it sends. Same thing for this one, Alt-C. Control is a caret. So the exponentiation sign is used to represent the control key, and so we're going to move through that particular script. So I'll run this script and you'll see how it works. So first of all, it says run the script, and nothing happens because we're not on GastroPlus. But as soon as I go to Gastro, then it says it's going to send the Alt-Y key to show the physiology tab. Now it shows the physiology tab. Next, it'll show the compound tab. Next, it's going to go to move to the next record, move to the next record, and start a simulation. Now, I didn't press any keys. I mean, Autoit is handling the entire process, but you could imagine setting up a iterative process in Autoit that would demonstrate and help you automate those processes. Okay, I think I'll stop there and we'll move forward.
Thank you, Mike. So in the interest of time, I think we're going to move on to the poll and then get into the Q&A. So Arlene, could you please bring the second poll? Oh, I'm sorry. Yeah, the take-home messages. So the gurus, could you please very quickly, 30 seconds or less, go through your take-home messages?
Sure. So Michaela first, although GastroPlus does provide valuable tools for modeling and simulation, let the buyer beware. You have to be cautious to use it responsibly and appropriately. And sensitivity analysis is one important piece for that to identify potential weaknesses and areas of uncertainty for your model.
And the next item was regarding the biorelevant solubility calibration. It's always a good idea to verify that the predicted solubility that GastroPlus is using matches up with the measured solubility. And when it doesn't, you can do some tricks to try to make it do so.
So the next point is probably from the colonic absorption settings, but I guess it's pretty much beyond that. I would say the PBBM is a data-driven process, so it is critical to check and refine your models appropriately at different stage of development using available data with properly designed experiments for parameterizations of key aspects of the model.
I would say, pay attention to the capability of these keyboard overrides. We're going to try to build a lot of these into the GPX program that's coming out hopefully by the end of the year. And consider, automating processes that normally would take you a long time. Maybe you've got some IVIVC optimizations and you want to start that and just, go home and come back in the morning.
All right, thanks again, all the speakers. So Arlene, could you please bring up the poll? So I'd encourage everybody to please respond to this 30 seconds before we get into the Q&A. All right, thank you, Arlene. So we're going to get into the question and answer session of this webinar. We had, as I had mentioned up front, we had quite a few questions posted prior to the webinar, so we'll get started with that, but I already saw a few questions that were asked through the question box, so I appreciate those. But let's get started with the questions that folks had posted prior to the webinar. So the first question, which is pretty detailed, but I will just summarize that it's a question on a recommendation for simulation of amorphous solid dispersion. So Mike, would you take a first stab at that? And then I will ask Phil to chime in too.
Actually, let's let Phil, answer first, and then I'll describe some of the newer things that are available in 9.7.
Okay. So at Vertex, we've built a lot of models of amorphous dispersion. It's one of our almost default formulations we use preclinically and getting ready for FIH. So my basic practice is to use the particle-based dissolution model, so the standard model that shows up when you open up a new database. And the trick with amorphous dispersions is getting the right solubility inputs. So my approach there is always to first build a model of the crystalline form solubility. So similar to what I presented, you need the pKa's measured directly. you need solubility in a buffered system where you know the pH, and you need the solubility in biorelevant media. And then the next step would be to get the amorphous solubility. It's just an increase in the intrinsic solubility. So you could measure the solubility of the amorphous and crystalline forms in the same media, and the ratio of the solubility is just a correction factor that you can apply to the solubility to represent the amorphous dispersion. All of the other parameters that you've built with the crystalline model will be the same. And then with the new version of GastroPlus, which Mike can explain in a little bit more detail, there's an ability to model two different solid forms. One can be amorphous, and the other the most stable crystalline. So with that, I'll turn it over to Mike to explain that.
So maybe just make me the presenter for just a couple of seconds here. A lot of you have probably taken the course that we offer in the workshop, and we run through an example of a solid dispersion for nimodipine, which has 3 different doses, 30, 60, and 90 milligrams. The classical nucleation and growth theory says that precipitation times will increase as the dose increases, because the level of supersaturation is going to be increasing with each particular dose. The whole idea of an enabled formulation is to drive as much of the drug into solution as you can, as fast as you can, and then let whatever happens from precipitation take over. The latest incarnation of this in GastroPlus 9.7, we've added the ability to increase the number of molecular forms that are moving through the small intestine. And so when you see the reference solubility, which is the aqueous solubility in most cases, and you click on more solubility, you have the ability to specify which form of the drug you want to administer. In this case, it would be 100% of the amorphous, which has a solubility that is 10 times higher than the aqueous solubility. And then we checkbox the solubility for the precipitated particles and say that the precipitate will be the crystalline material and come out with a solubility that's 10 times lower than the amorphous solubility that was administered. The beauty of this is that in the past, we typically would drive the drug into solution with a Weibull function that was very fast, and it would go up to 100% and then end up with some precipitation. The nice part about the new format of this two-form solubility is that you can use the Nernst-Bruner equations to drive the drug into solution based on the particle size of the administered form. So in the case of the amorphous material, it would undoubtedly be a smaller particle size than you would see for the other forms. And so when you are looking at your solubility, you can basically set up all of the individual characteristics for the different forms as far as particle size goes. We can, check this out at our next workshop, where we'll be over in Germany, in Mainz.
Thank you, Mike and Phil. So moving on to the second question, it's about the best practice for estimating permeability. And there are two scenarios rather. One would be prior to any clinical data available, and the second is after we have some first-in-human data. So I'll ask Phil to maybe chime in first and weigh after that.
Sure. Preclinically, at least at Vertex, I believe we use a standard approach, which is to use a cell-based assay to get the in vitro permeability of a compound. And then by measuring the in vitro permeability of a number of control compounds where we know experimentally the human jejunum permeability, you can use GastroPlus to build a correlation for your specific lab's assay. And so we use that type of approach to get the initial estimate for the permeability. If we have preclinical data, say in a rat or a dog with a solution formulation, that can be also quite useful for either verifying and maybe sometimes optimizing the in vivo permeability based on that initial in vitro input. And then maybe I'll turn it over to Wei if he wants to add anything.
Yes, thanks, Phil. So I would say it's pretty much relatively similar in Merck. We actually have the cell-based assay, and also we have rat intestinal perfusion database as well. So basically, with this database, we have internal correlations of the permeability from either models to the humans. So this is one part, and I guess I totally agree with the fields about the solution formulation, IR, oral data, which might be useful as well for optimization of the model. And one thing that I would say that just to get to the question that I guess what would be the best practice on these two scenario, option A and B, is I would say that it's kind of representing the different purposes of the absorption model at different stage. So I would say in human, those projections, formulation effect in early stage of the development, at that time we actually didn't really have the human data. So we will have to rely on the cell-based or in situ-based models. But later on, we certainly will need to refine the model. I think that's kind of like the standard practice in Merck during the late stage in silico work for the formulation bridgings and issues once we have the clinical data.
Thanks, Wei and Phil. So moving along. So next question, and I would pose this to Wei, and the question is about prediction of oral absorption in peds and elderly population. The question is a bit broad, so maybe from the way I read this is if you can talk about some best practices around that and your or Merck's overall satisfaction with these predictions.
Well, I guess overall, I think it's a good question. It also represents one of the challenging areas of the PBPK, PBBM, and beyond, which is kind of like PBPK modeling. So I would say for pediatrics, especially for younger age kids, which PBPK modeling can potentially play a bigger role in those estimations, those optimizations. From absorption perspective, I think we will need to consider the factors such as age-dependent change in gastric pH, bile salts, etc. And also, I think one important factor is frequent feeding intervals on infants, and I think that would likely impact the effective GI transit time and pH as well as hepatic blood flow. So these should be considered in your model as well. I guess overall the literature data has been relatively limited, but there are several manuscripts on investigation of the age-dependent GI physiology, including pH transit time, GI fluid characterizations, and GI fluid volumes using physical-chemical characterizations of clinical samples and also the imaging techniques. So I think these probably may be interest for people working in this area as well. For the geriatric side, I don't have extensive experiences, but I think literature reported that the dissolution of the drug is pretty much affected by the aging of the GI mucosa in the elderly and the decrease of the GI acid secretions. So although these effects, I think, maybe at least partially offset due to the weakening of the motility of the GI in the elderly which resulted in a slower gastric emptying rate. But overall, I would say that if a drug is a weak base with steep pH solubility profile, theoretically, I think one would expect a lower and a more variable PK in elderly than in adults due to impaired dissolution and absorption of the drug. So these physiological factors will need to be considered when you build the geriatric absorption models, and of course, refinement with the clinical data in the elderly.
Thank you, Wei.
This is Michaela. I have
next question.
A little bit more to add there.
Sure.
So
go ahead.
I've actually, thank you. I've actually been involved in some simulations with GastroPlus for neonates, and we had some success there. But before this Guru's discussion, I talked a little bit with Vera to kind of find out how much data went into GastroPlus. And for the neonates and the pediatric population, she told me that there were a lot more data available for height and weight and the PBPK piece and less for the GI tract. So especially as the ages get younger, you have to be cautious, and the data are just limited for the physiology affecting absorption modeling. And the other piece I touched bases with her on was the elderly. And she said that the model basically does change the height and weight, and so the PBPK piece reflects the size of the elderly population and the liver volume, but there were limited or no changes to the intestines. So, for example, if you're concerned about an acidic elderly person, that would be a stochastic thing anyway, because some elderly people have, stomach acid. But you would have to actually go in and adjust that in the ACAT model yourself. Thank you.
Thanks, Michaela. So next question is actually for you. So the question is it advisable to use PK plus generated Ka values from compartmental fitting and calculate regional effective permeability? And the second part is, if there is no effect, if experimental effective permeability data are not available, then what would be a recommendation? I guess that kind of goes into the second question, but still.
Yeah, I would suggest being very careful with that. I mean, if you don't have any other option, then you kind of do what you can and just make sure that you try to check against more in vivo data if possible. I personally, in that situation, would do as much literature searching as I could to try to find cell-based data in the literature if possible. Also, maybe look for compounds with a similar molecular structure in the literature, if that's a possibility. And with the scientific method, we try to look at more than one approach. I might also use an in silico P effective, or at least look at it and calculate the in silico P effective and see if it's a high permeability molecule, in which case an exact value isn't that important, or if it's a lower permeability molecule, in which case you might want to consider more carefully whether you'd actually have to measure it before doing your modeling. So if you can find a couple of different pieces of information and it all kind of comes together, then maybe you could use this approach. But I'd just be cautious and try to look at other alternatives and do some sensitivity analysis and really think about your BCS class and whether your Ka value is reflecting permeability or maybe dissolution or some other process. So thank you.
Thanks, Michaela. So I will now go maybe on the live chat here. So Franco, you had your hands on for quite some time now. So please go ahead and ask the question.
Actually, that's okay. I did it by mistake and I tried to reverse it, but apparently it doesn't do that. Okay. That's okay. You can skip that. Thank you.
All right. Thank you. And I see Muhammad Faraz. You have your hands up. You have a question? Okay, I guess not. Darwin Cow. Should we unmute now to ask your question?
Can you hear me now?
Yes.
Yeah, the question is for Phil. You mentioned when you do the fast state versus fastest state simulation, right? Obviously, the solubility is affected by pH. And I thought, have you had a situation where I have seen is, for example, you have a weak base, right? And the solubility, obviously, in the default GastroPlus simulation, in the gastric pH is quite high. It's about pH 4 and 5. And that can result in a prediction of a negative food effect. And in reality, sometimes, I think what we have seen is if we change the pH a little bit, because during the longer stomach emptying time, the pH eventually comes down. And that is more consistent with the clinical results. Have you had to make the adjustment for the fast state gastric pH?
Yeah, that's a good question. I know it plagues everyone working on weak bases, and I've had certainly cases where, yeah, the gastric pH, because of the difference between fasted and fed, will predict a negative food effect for a weak base, and obviously the clinical data usually tell a different story. I guess I wouldn't be inclined to change the physiology parameters unless I had a solid reference on which to base it, particularly if it was say a regulatory filing or I needed to convince someone with actual data about that. But I think that's kind of an open question. It might be something to pursue in the future. Yeah.
This is Mike. In the case that you're talking about, you just mentioned weak base, but you didn't say exactly what the pKa would be. And there's a big difference between a pKa in the 4 to 6 region versus the 8 to 10 region. In one case, the solubility is going to change with regard to the increase in gastric pH. In the other case, the solubility won't change very much at all. So, each one of these specifications is important. And one of the things that happens in a typical kinase inhibitor with a pKa in the 4 to 6 region is that the formulation becomes dependent on particle size, whereas in the fasted state with low gastric pH, it's not. So chemistry classification is an important characteristic in this situation.
Yeah, thanks, Mike. The specific example I was thinking about, the pKa is 3.7, so that did have a pretty big effect.
Right, and in those situations when the fed condition has a increased gastric pH, then the formulation does become dependent on the particle size because of the low solubility in the gastric environment.
And I have one other thing to add, is you might want to check out what the salt plateau is for the sweet base. If you are dosing it as a salt, just to make sure that salt plateau is at the experimental solubility of the salt. If you use the default values, they could be significantly off.
Thank you.
Thanks, Phil and Mike. So Mike, the next question is for you, and it's actually on the OCAT model. So we have been primarily talking about oral, so I thought it was interesting to go into ocular. So the question is twofold. So it's future plans to add an ocular lens compartment to the OCAT module, and then I'll go on the second one after I answer this.
Well, we've been talking about adding an ocular lens compartment to the OCAT model for several years, actually. We've had a grant from the FDA for ocular that ran for 3 years and it was then extended. And no, we haven't added an ocular lens compartment yet. This is one of those lower priority items that may be important or significant, and I know the group that is asking the question, and certainly we can discuss that and maybe give it a higher priority.
Thanks, Mike. And the second question was on the future plans to double the number of atoms capability in an imported structure.
Sure. I mean, that's a 10-minute coding change that could easily be accommodated. So anybody who is interested in some of the newer biologic applications, antibody application to the eye, peptide application to the eye, siRNA application to the eye. A lot of these may have some artificial limitation in GastroPlus. Just let us know about it, and we can make a quick change on that. And anybody interested in cyclic peptides, contact me, and we can have a discussion, because it's high interest right now.
Thanks, Mike. So we have, again, quite a few hands up right now. Sandeep? You are on mute.
Hello.
Yeah, we can hear you.
Yeah,
Go ahead.
I have a question that if IV data in human is not available, can we take animal IV data and extrapolate to humans?
This is
Who wants to take that?
Michaela. I mean, you can, but I would be very cautious and do all kinds of uncertainty analysis. And I mean, if you can look at in vitro data, one approach that, I'm sorry, I'm hearing an echo. If people could mute their phones, thank you. So one approach would actually be to use PBPK if you had in vitro data, and then you could use extrapolation of the IV and compare the PBPK and see if they're similar, and that might raise your confidence. So I think the answer is yes, but you have to be very cautious.
All right, thanks. Saima?
Can you hear me?
Yes, go ahead, please.
So I have a question regarding how can we use MDCK MDR1 generated permeability data and use it to predict the human effective permeability, because I don't see that module in GastroPlus. I see CACO2 and other cell lines. So can you comment on that? Thank you.
This is Mike. Yeah, just if I could answer this one. We have a program called Membrane Plus. This is a in vitro simulation of the cell culture permeability experiment. We also have suspended hepatocytes and sandwich culture hepatocyte models built into Membrane Plus. If you would take your MDCK MDR1 permeability data, particularly if you have the kinetic data rather than just the final permeability experiment, and put that into Membrane Plus, you would be able to develop a correlation that would couple over to GastroPlus that would give you an effective permeability that you could use quite nicely. It's just a matter of calibrating with some of the known BCS standards.
Thank you, Mike. So we have less than 2 minutes left, so I think we should close the webinar now, but we still have a lot of questions that are unanswered, so I apologize on behalf of the group that we couldn't get to all the questions. But what we could do is, folks here are probably familiar with LinkedIn GastroPlus user group, so I would recommend that you post the questions on the user group, and we will respond to them quickly. So with that, I just want to take a minute to acknowledge the user group steering committee for organizing this webinar, especially Michaela for spearheading this effort. Simulations Plus for helping us host this, particularly Arlene, who worked quite a bit in the background on all the logistics, and also MS-DOS early Windows. And as I mentioned, there is a LinkedIn user group. Please join if you're not part of that, and be active users and post your questions on LinkedIn, please. So with that, thanks again to the 4 speakers for sharing your experience with us today. And with that, we'll close the webinar, and thanks everybody for calling in and participating. Thank you.